Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic terms. And our motto is 5 terms at a time, keep the learning in the mind. The first most common term is centromere. The centromere appears as a constricted region of a chromosome and plays a key role in helping the cell divide up its DNA during division, that is mitosis and meiosis. Specifically, it is the region where the cell's spindle fibers attach. Following the attachment of the spindle fibers to the centromere, the 2 identical sister chromatids that make up the replicated chromosome are pulled to opposite sides of the dividing cell, such that the 2 resulting daughter cells end up with identical DNA. Picture a chromosome in your mind. Even if you've never actually seen one under a microscope, I bet you've pictured sort of an X shape, where the arms of the X are wider and there's a thin part at the middle where they all join. That thin part is the centromere. The centromere is not always in the middle of the chromosome. In fact, sometimes it can be almost all the way at the top. That is called an acrocentric chromosome. What you can't see is the very special kind of DNA that makes up the centromere. It is a specific kind of repetitive DNA called alpha satellite. The alpha satellite DNA is where the proteins that the centromere needs to attach to the spindle fibers come together. The second most common term is chromatid. A chromatid is one of the 2 identical halves of a chromosome that has been replicated in preparation for cell division. The 2 sister chromatids are joined at a constricted region of the chromosome called the centromere. During cell division, spindle fibers attach to the centromere and pull each of the sister chromatids to opposite sides of the cell. Soon after, the cell divides in 2, resulting in daughter cells with identical DNA. Cell replication is important during growth and for replacing cells that are damaged and cannot be repaired. A good example of this are the cells that are in place when you get a cut on your skin and the cut heals. The new cells are exact copies of the old cells. As part of cell replication, your genetic material that makes you who you are, which is found in your chromosomes, must be copied and directed to each of the new cells. These chromosome copies are chromatids that have a special structure that connects the 2 and allows for this careful separation into new daughter cells to maintain a correct number of chromosomes in each cell. The third most important term is chromatin. Chromatin refers to a mixture of DNA and proteins that form the chromosomes found in the cells of humans and other higher organisms. Many of the proteins, namely histones, package the massive amount of DNA in a genome into a highly compact form that can fit in the cell nucleus. The total DNA in the cell is about 5 to 6 feet long, which has to fit inside the nucleus of a cell in an orderly fashion. DNA molecules first wrap around the histone proteins, forming beads on string structure called nucleosomes. Nucleosomes further, inaudible, and condense/gather to form fibrous material which is called chromatin. Chromatin fibers can unwind for DNA replication and transcription. When cells replicate, duplicated chromatins condense further to become a lot like chromosomes, visible under the microscope, which are separated into daughter cells during cell division. The fourth common term is chromosome. Chromosomes are thread-like structures made of protein and a single molecule of DNA that serve to carry the genomic information from cell to cell. In plants and animals including humans, chromosomes reside in the nucleus of cells. Humans have 22 pairs of numbered chromosomes, that is autosomes, and 1 pair of sex chromosomes, that is XX or XY, for a total of 46. Each pair contains 2 chromosomes, 1 coming from each parent, which means that children inherit half of their chromosomes from their mother and half from their father. Chromosomes can be seen through a microscope when the nucleus dissolves during cell division. Chromosomes vary in number and shape among living organisms. Most bacteria have 1 or 2 circular chromosomes. Humans, along with other animals and plants, have linear chromosomes. In fact, each species of plants and animals has a set number of chromosomes. A fruit fly, for example, has 4 pairs of chromosomes, while a rice plant has 12 and a dog 39. In humans, the 23rd pair is the sex chromosomes, while the first 22 pairs are called autosomes. Typically, biologically female individuals have 2 X chromosomes, that is XX, while those who are biologically male have 1 X and 1 Y chromosome, that is XY. However, there are exceptions to these rules. Chromosomes are also different sizes. The human X chromosome is about 3 times larger than the human Y chromosome, containing about 900 genes, while the Y chromosome has about 55 genes. The unique structure of chromosomes keeps DNA tightly wound around spool-like proteins, called histones. Without such packaging, DNA molecules would be too long to fit inside cells. For example, if all of the DNA molecules in a single human cell were unwound from their histones and placed end to end, they would stretch 6 feet. The last fifth term for today is cloning. Cloning, as it relates to genetics and genomics, involves using scientific methods to make identical, or virtually identical, copies of an organism, cell, or DNA sequence. The phrase, molecular cloning, typically refers to isolating and copying a particular DNA segment of interest for further study. Cloning is a word we use to describe a laboratory process of making millions or even billions of copies of a single molecule, most often DNA. It's different from the uses of the terms cellular cloning or organism cloning that are used in reproductive genetics. We use molecular cloning to amplify or make many copies of DNA, which allows us to study the molecules in the laboratory, and to perform genetic tests on small samples of blood, saliva, or other tissues from patients. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.